Aloha everyone, and welcome to Unit 3 here at Chaminade University for Biology 101. Today we're going to discuss biological molecules, so the different types of molecules that make our cells run, basically. So we talked previously about atoms and how atoms interacted, and that gave us a little bit of a clue of how carbon was so important. Remember, it's able to make 4 separate bonds, so it can make either 4 single bonds or 2 double bonds or a double bond and 2 single bonds, and that allows it to have a lot of flexibility in the creation of these organic molecules. And organic molecules are going to be carbon chain-based molecules. We're going to look at 4 major types of these molecules: carbohydrates, lipids, proteins, and nucleic acids. And this is going to go hand in hand with the laboratory session that we're going to have on biological molecules. So before we get started, I want to talk about something called monomers and dimers and polymers, and I know we'll get to that in a little bit later, but each one of these different types of biomolecules have a monomeric form, a dimeric form, and a multimeric form or a polymeric form. And that means that we have something like you could consider an individual train car, and then multiple train cars linked together, and then the entire train. So the monomer would be a single car, dimer would be 2, and a polymer would be many of the train. And each one of these will have different bases for the individual train car, but they're all going to be able to link together. And we'll talk about how that works in just a second. But let me back up for a second. We're talking about organic molecules. And organic is different from a scientist's perspective than it is from, say, a vegan's perspective, right? Organic simply means that it's a carbon skeleton with hydrogen atoms on there. And inorganic is anything that is without a carbon, or something like carbon dioxide, which only has 1 carbon. So carbon skeleton has to have 2 or more carbons. And what's interesting, right, any vegan that tells you that they only eat organic, you can go ahead and challenge that and tell them that water is an inorganic molecule, and everybody needs water to survive. So, all right, moving on. So, carbon, as we mentioned, is very important. It's able to make 4 separate bonds, and that's because of the electrons and what we call the valence shell. And for reference, you can head back to Unit 2 when we talked about atoms to figure out a little bit more about that. But the point is that carbon atoms are able to make 4 bonds each. So they can make a triple bond and a single bond. They can make 2 double bonds. They can make 4 single bonds. And so they can make multiple different shapes, which allows us to create things like chains, rings, sheets, and even helices. So just to show you how carbon is unique compared to the other atoms in terms of the versatility, carbon here is able to have 4 separate sets of bonds, whereas hydrogen is only able to have 1. Nitrogen, for reference, is able to have 3. That allows it to make either 3 single bonds, or a single and a double, or 1 triple bond. Oxygen is a little bit more restricted. It can make 2, so it can make 2 single bonds, or 1 double bond. Carbon, however, has more versatility because it's able to make 4 single bonds, or as depicted here, 1 double bond and 2 single bonds, or 2 double bonds, or a triple and a single. So my point is that there are more orientations for the carbon atom to interact with other atoms to be able to make different shaped molecules. And that's going to be very important for all of the different biological molecules that we talk about, because that gives it a lot of complexity and different unique shapes that we're able to create and interact with one another. Speaking of interaction, functional groups are going to be reactive groups. These are going to be groups that are going to determine how that molecule interacts with the molecules around it. So functional groups tend to be reactive. That means they're a little bit less stable than something, say, the carbon backbone, which is generally very stable, and are going to be the parts that are going to be participating in the chemical reactions with other molecules around them. I do not expect you to memorize this, but I want to just show you that we have multiple different types of biological molecules and different groups that are going to hang off of these biological molecules that afford them different properties based on their structure. All right, so a hydroxyl group we'll talk about quite often. That's an O and an H. This is a polar molecule, and it's going to be involved in what we call dehydration synthesis and hydrolysis reactions, and we will talk extensively about that throughout this chapter. This is basically how molecules are going to be built up and broken down. The hydroxyl group is found in things like sugars and polysaccharides and amino acids. Now, I'm not going to go through each one of these individually, but I do want to talk about the carboxyl group and the amino group for a moment, because these are going to be found as the reactive groups for amino acids, right? So an amino acid is going to have one side that's an amino group and another side that's a carboxylic acid group, and these are going to interact with the molecule next to them. So one amino acid's amino group and the second amino acid's carboxyl group are going to interact with each other to form a peptide bond, which is going to help us put together amino acids into peptides into proteins. All right, a phosphate we're going to talk about extensively when we talk about ATP. So you're going to see 3 of these in a row in ATP and only 2 in a row in DP. So tri would have 3, di would have 2. It's the high energy bond in ATP. So you'll see ATP to ADP used a lot throughout the rest of this semester. And when you think about that, you're going to think about this phosphate bound to another phosphate, bound to another phosphate. Another place you're going to see this is in the sugar phosphate backbone that you're going to find in DNA. And we'll talk about that a little bit later on. DNA gets its own chapter though, so we won't go too heavily into the structure just yet. All right, as I mentioned, the smaller organic molecules, the very smallest form of that organic molecule that still keeps its form as a molecule, is called a monomer. And we put them together, a 2 together makes a dimer, 3 would make a trimer, and as they get longer and longer, they're simply called a polymer. And these are created by dehydration synthesis or hydrolysis. So what do I mean by that? Basically what happens is when we have 2 monomers, picture 2 individual train cars, and they have to get joined together. And the linkage site where they're going to get joined together has to kind of be opened up. So what happens there is that one hydrogen is going to come off of molecule A, and a hydroxyl group is going to come off of molecule B, and that's going to allow a little bit of reactivity between those molecules, and water is going to be lost at that point. Right? And once water comes away, now we have a little bit of positive on one side, a little bit of a negative on the other side. That creates a covalent bond between those 2 subunits, and we go from a monomer to a dimer, or from a dimer to a trimer, etc. What does that actually look like? Okay, so here we have molecule A and molecule B, right? We have the loss of this hydrogen and the loss of this hydroxyl group are going to create kind of these sticky ends here with this oxygen. These 2 together are going to create a water molecule. So that's a dehydration where we're pulling water away, and we're synthesizing these molecules together into a dimer. So that's why it's called dehydration synthesis. You can see what happens is that oxygen molecule is going to bind from molecule A and molecule B, and now we have a dimer instead of previously having 2 monomers. And while this can happen in this direction, right, as you remember previously, anytime that you have a reaction that's going to build up molecules or put molecules together, it's anabolic. Think anabolic steroids pump you up, right? And anytime that molecules are broken down, it's catabolic. Picture what the cat likes to do with the vase is break things, right? So the process of the 2 of these together is metabolism. But my point is that pretty much any time that we have an anabolic reaction, that we're building things up, it's going to be through the process of dehydration synthesis. And the reverse reaction, when we are breaking things down, is going to happen through hydrolysis. So remember, we just built that water molecule. The equal and opposite reaction will be breaking that water molecule down, or water cutting. And then that that hydrogen and the hydroxyl group are going to end up being used to kind of cap off the ends between what was a dimer that is now 2 monomers. And this is the equal and opposite reaction that I just showed you. So that water molecule is going to get split in half. That's why it's called hydrolysis, hydro being water, lysis breaking, right? And when that happens, this dimer can be turned into 2 monomers because this oxygen is happy here with the hydrogen, and this OH group is going to serve to make this molecule happy as well. So we just had a catabolic reaction where we took a dimer and broke it down into 2 monomers. And this is going to happen with pretty much all of our biological molecules that we're going to discuss going forward. All of them are going to be created by dehydration synthesis and broken down by the process of hydrolysis. As I mentioned, we have 4 major groups of biological molecules. The first are carbohydrates, the second are lipids, next proteins, and then nucleotides and nucleic acids. And we're going to touch on all of these throughout the rest of this chapter. Now this table summarizes everything that I'm going to tell you, so I'm going to kind of gloss over this and then we'll dive into it in depth in the following slides. But again, we have 4 major types of biomolecules, and here it's listed the type and also the structure of this molecule. So carbohydrates are going to be primarily in the form of CH2O, little parentheses here, to the N power. And what does that mean? It basically means that anytime you see a molecule that, for example, this would fall into it, C6H12O6, right? If you have double the amount of hydrogens as you do oxygen and carbon, and it's probably going to be a monosaccharide of a simple sugar. So monosaccharides, again, monosaccharides are going to be the train car of a sugar, just one train car, right? And some examples of these are going to be glucose, fructose, or galactose. Again, these are monomers of sugars. Now when we take 2 monomers together, we're going to make a disaccharide. In this case, the monomer would be a monosaccharide, so it's a specific type of monomer, saccharide being sugar. So 2 monosaccharides together make a disaccharide. 1 more would be a trisaccharide. Eventually we get to polysaccharides, so a whole chain of those monomers of sugars or monosaccharides. And some examples of that are going to be starch, glycogen, and cellulose. When we talk about the lipid section, we're going to talk about the very structured carbon and hydrogen backbone. It's a very high proportion of carbon and hydrogen. Not very many other things are going to be in there, and it's actually going to be insoluble in water. Picture something like bacon grease or vegetable oil. Those are going to be lipids, and they're going to be nonpolar. Remember, water is a polar molecule, and nonpolar and polar molecules don't interact well, just simply like water and oil. And we have multiple different types of this, including a triglyceride, waxes, phospholipids, which we'll talk about extensively when we talk about the plasma membrane, and then steroids, which are used to regulate hormonal function and also are something that's actually a lot more tame, like cholesterol. Proteins. So proteins, again, they're going to have the monomeric form and the dimeric or polymeric form. The monomeric form is a peptide, is an amino acid. The dimeric or more is going to be a peptide, so that's a chain of amino acids put together, and then a long chain of those would be a polypeptide, so a long chain of amino acids. And we'll talk about some examples of proteins and protein function when we get to that point, but proteins are the workers of the cell, so they do almost every function that you can think of within the cell. Lastly, we'll talk about nucleotides and nucleic acids. Now, nucleotides and nucleic acids are going to be the backbone of your RNA molecules and your DNA molecules, but also the backbone of your ATP molecules and some other energy carrier molecules here that we talk about cyclic A and P. But again, the main place that you're going to find them is going to be in your DNA and then all of your ribonucleic acids, which are going to include your mRNA, your tRNA, and your ribosomal RNA. All right, so let's talk about carbohydrates first. And monomers of carbohydrates are always composed of carbon, hydrogen, and oxygen at a 1 to 2 to 1 ratio. Again, a monomer of a carbohydrate is a monosaccharide, 2 together a disaccharide, 3 or more, we would go trisaccharide into polysaccharides. Carbohydrates are very important because they're a high energy source for organisms, and they're generally going to be water soluble. If you picture that you put a spoon of sugar into a glass of water, it will dissolve, and that's because it has a polar group, an OH functional group hanging off there, and again, polar molecules dissolve well in a polar solvent like water. So the backbone of sugars is going to be 3 to 7 carbon atoms long. Glucose you may know as 6. And they're going to have a hydrogen group on one side and a hydroxyl group on the other end, and this is going to allow it to make a backbone, so it's going to be able to bond to the next sugar molecule beside it. Again, we have an approximate chemical formula of double the amount of hydrogens as we have carbon and oxygen, so 1 to 2 to 1, and N would be just whatever the number of carbons is, we would double it for hydrogen and keep it the same for oxygen. Again, C6H12O6 being glucose. Again, generally these are going to be able to dissolve in the cytoplasmic fluid because cytoplasmic fluid is generally a waterous or aqueous environment. And when it's dissolved, it's going to form a ring. So we're going to depict carbon generally as a 6-carbon, we're going to depict glucose or sugars as a 6-carbon ring. Some of them are going to be pentamers and 5-carbon rings. And glucose is going to be the most common monosaccharide that we're going to talk about. So you're going to see it depicted here. This is a space-filling model, but you're generally going to see it depicted here in a simplified ring version where you're just going to see this here is a hexamer, but sometimes other sugars are going to be pentamers, but glucose is a hexamer, which means it has 6 sides. There's multiple ways to depict these different molecules. So for example, all of these depict the molecule glucose. I'm not going to ask you to memorize this, but just to demonstrate the fact that we have multiple ways of showing the same thing. This here is a chemical formula for glucose, again C6H12O6, and that's just telling you the number of each of the atoms that are found in the molecule, but it's not telling you the arrangement or the order or how these guys are actually aligned up. This is a ball and stick model which shows you the linear form of the carbons. You can see they're depicted as 1 through 6. Again, it's a 6-carbon ring, but actually when it makes a ring, one of the carbons kind of dangles off, and we have a link between this carbon that is bound to oxygen for a second, and that's going to be the area that's going to be reactive. And if we simplify this version down, we're going to see something like this. And again, we're going to have our carbons labeled 1, 2, 3, 4, 5, and 6, so most of the carbons aren't actually going to be depicted. And from here on out, you might just see it depicted as a hexagon. All of these are going to be the molecule glucose. But glucose isn't the only monosaccharide. We have other monosaccharides like fructose, which you might find in your corn syrup, for example, in your honey, and also in your fruit sugars. We also have galactose, which is a milk sugar, and ribose and deoxyribose, which are going to be very important when we talk about RNA and DNA. So we were just showing previously that glucose is a hexamer. It has 6 sides. Here's fructose, an example of a pentamer. It has 5 sides. And I'm not going to quiz you on any of this. I just want you to show that if you see a series of pentamers and hexamers together, you know that you have a series of sugar molecules that are bound in a long chain. This is what ribose and deoxyribose look like. Again, I'm not going to quiz you on this, but the difference being that there's the loss of one oxygen. That's right here. And so that's going to be a very interesting point when we talk about the RNA being allowed to travel out of the nucleus to be able to bind to the ribosomes and create proteins in the cytoplasm. Deoxyribose, however, is not going to be allowed out, so a DNA molecule, which is comprised of this sugar, is going to get stopped by the nuclear pore gate guards, if you will. Now, what's the point of breaking down molecules and building up molecules, right? And eventually the fate of all monosaccharides is that they're either going to be broken down or used for storage, right? So we're going to either break the sugar down into the energy that we're going to use to be able to drive all of the chemical reactions inside the cell, or if we eat more than we're actually using in our energy consumption, we're going to start linking the extra ones together via the process of dehydration synthesis for storage for later on usage. Disaccharides are, again, they're dimers, so they're 2 sugars put together, and they're going to be our short-term energy storage. They basically are going to be broken down into monosaccharides pretty easily by the process of hydrolysis, and that's going to be something that we can then use pretty much immediately, similar to a monomer. It's only one step removed from that. Again, this is the process of dehydration synthesis, whereby we are taking 2 monomeric sugars, in this case glucose and fructose, and we are combining them through the process of dehydration synthesis into this dimer. It's called dehydration synthesis, again, because we are taking the hydrogen and the hydroxyl group, bringing them together, and creating this water molecule. There are several examples of disaccharides, or two sugar molecules, including sucrose, which is glucose and fructose, lactose, which is glucose and galactose, and maltose, which is glucose and glucose. And anybody who's ever enjoyed a Whopper malt ball has tasted the effect of maltose as opposed to lactose, which is, again, the sugar that you might find in milk. This is something that some people are intolerant to because they're unable to create a very active version of lactase, which is the enzyme that breaks down that milk sugar lactose into the monomeric versions that you're able to digest properly. All right, so we have a disaccharide that ends up with a couple more monomers added to it. Then we end up with a polysaccharide. So it's a long chain of monosaccharides, 3 or more, right? 3 would be a tri. Anything higher than that would be a polysaccharide. And these are for storage generally. So things like starches. And starches you're going to be able to find in most plants. So things like rice, right? So, or potatoes. These are all found in, those are tubers. So these are high starch molecule or high starch foods. From the animal, you can get starch from the liver and the muscles in the form of glycogen. Glycogen is an energy storage molecule found in animals, and it's another type of storage polysaccharide. Both starch and glycogen are long polymers of glucose molecules. So what does this look like? And I told you going forward you might see sugar molecules depicted as just hexamers here. So these are starch grains that are found inside a potato cell. And starch molecules have they have specifically ordered chains that are actually cross-linked at specific locations, so that before we're able to get to each one of these individual glucose molecules, when we're breaking them down, first we're going to have to break these long chains too. So it's going to be a longer-term storage molecule for energy. But some organisms are going to be using polysaccharide also as a structural material. So for example, glucose is one of the most common versions of a structural polysaccharide. It's found in the cell wall of all plants, and most animals are unable to digest it. So for example, this is what surrounds the kernel of corn, and this is why for elderly people who are unable to chew properly, we've pretty much always in several societies found a way to grind the corn into cornmeal or make creamed corn or something where we actually break open the cellulose so that the digestive enzymes are able to get inside to get to the inside of the corn. What animal is able to digest cellulose? Anybody know? That's going to be termites. So a termite is able to digest the cellulose found in wood. So one of my favorite dad jokes that nobody ever laughs at goes a little bit like this. So a termite walks into a bar and asks, "Where's the bartender?" And while it's not funny, it exemplifies the point that the termite is actually thinking of the bar, as the wooden bar, as food. So cellulose is found here in wood, and it's specifically found in the cell wall of all plants. This is what that looks like up close. And you can see we have cross-linking of the long chain of monomers, which becomes a long polymer. that is an alternating configuration that we as humans and other mammals are unable to digest. But termites have a specialized enzyme that is able to get in there and cut those cross-links, so they are able to access that glucose. Another example of polymer is chitin, and if you've ever seen a crab or a hard-shelled insect or hard-shelled spiders, another example is fungi will make chitin walls around the outside, and this is a very strong polymer of glucose units that's going to basically be, well, what you consider to be a shell. Very strong, and it has these specialized regions that hang off of it that are going to allow it to create this conformation that makes it very rigid. All right, so moving on from carbohydrates, we're now going to switch gears and talk about lipids. Lipids are another specialized type of biological molecule. They have long regions that are basically long hydrocarbon chains, just long chains of hydrogen and carbon. And they're generally going to be, again, a long chain. These are hydrocarbons that are non-polar. And remember, the water is polar, so lipids are not going to interact well with water. That means they are water insoluble, which is a term that we use, hydrophobic. You're going to hear hydrophilic and hydrophobic used throughout the rest of this class. Hydrophobic means water fearing, hydrophilic means water loving. So lipids do not interact well with water, they are hydrophobic. What's the purpose of lipids? Long-term energy storage, right? Fats, think of blubber in the whale, right? Also insulation, same thing, blubber in the whale. Also waterproof coatings. If you think the way the water goes off of a duck's back, that's because the feathers have lipids or waxes that are on the outside that help the water just run right off. Additionally, they're found in the cellular membranes, and this is going to be one of the most important things. We'll talk about this extensively in the plasma membrane chapter. It gets its own chapter. But additionally, they're also found as hormones, and we will talk about those right now, or in just a little short while. Things that are going to help regulate several of our biological processes. Now, the three major groups of lipids are all combined together into oils, fats, and waxes. Phospholipids, which again are going to form the structural components of the cellular membrane, and steroids, which are going to have multiple rings of carbon, hydrogen, and oxygen, and these are going to help regulate multiple biological processes, and also cholesterol falls into this category. Now, oils, fats, and waxes, although they seem different, what we would call phenotypically, like on the outside, they are made of similar subunits called fatty acids. And fats and oils are made from extra energy in the body. They end up being the energy storage molecules. So if you end up eating or bringing in more calories than you are actually expending, so if your caloric consumption exceeds your caloric expenditure, you can end up storing that in the form of fats. Fats are very important energy storage molecules because they contain nearly twice as many calories, over twice as many calories per gram as carbohydrates and proteins. So they're actually going to be, if you have 1 gram of fat, you're going to be able to get twice as much energy out of it. They're created by a process called dehydration synthesis. Who's surprised, right? Everything that's an anabolic reaction is created by dehydration synthesis. And it's the synthesis of 3 fatty acids plus a glycerol molecule equals something called a triglyceride. So what does a triglyceride look like? So here's your fatty acid chain. I'm going to take a second to talk about this fatty acid chain. It is a long chain, basically, of hydrocarbons. All right? It's basically carbon with 2 hydrogens, and each carbon is bound to 1 carbon on either side. Again, carbons can each make 4 bonds, so this carbon is bound to this carbon, that carbon, and 2 hydrogens, right? And then these fatty acid chains, 3 of them are put together to a glycerol molecule. Again, that's through the process of dehydration synthesis. It's going to make something called a triglyceride. If the triglyceride is saturated, what does that mean? That saturation means that the carbon chain has as many hydrogen atoms as possible, meaning that all of the carbon-to-carbon bonds are single bonds. And that looks kind of like this, right? All of these carbon-to-carbon bonds are single bonds. And that means that this is going to be a straight chain. If, however, again, this is going to be a straight chain. You can see this is a fat. If, however, they're unsaturated, that means we have at least 1 or more. 1 would be monounsaturated, 2 would be diunsaturated, past that polyunsaturated, something that has 2, or at least 1, double bond between 2 carbon molecules. And that might give a little bit of a kink in the tail. So this is what an oil looks like. And I want to go back a second to show you what that fat looks like. And the example that I like to use here are paper clips. So if you had a box of paper clips and they all lay flat, you could fit a lot more paper clips in the box than if some of the paper clips, or if all of the paper clips were kind of bent in different directions, right? It would be very difficult to fit all of the paper clips in that box. And that's the difference between oils and fats, right? The fact that these take up a little bit more space because they're expanded a little bit allows them to slide over one another a little bit more. That means oils are going to be liquids at room temperature, and fats, like bacon grease, for example, which are generally going to be saturated fats, or saturated chains of hydrocarbons, they're going to end up being solid at room temperature because you're able to pile more of them in a closer and more tightly structured lattice, basically. So that's the difference between fats and oils, is whether or not we have a straight fatty acid tail, which would be saturated, or whether or not we have at least one, if not more, areas where we have double bonds, which would make it an unsaturated fatty acid. Now, what are waxes? Waxes are actually highly saturated, and we talked about the saturation levels being affiliated with whether or not something is solid. And waxes are going to be very solid at room temperature. They're actually going to have a higher melting point even than the unsaturated, than the fats or the oils. And waxes are anytime you see a waterproof coating on things. For example, like I talked about water off a duck's back, but also any of the stems and leaves on the plants that are going to want to keep the water inside the plant cells themselves. Also, fur on mammals can get kind of waxy or greasy, and that's going to, again, allow the water to flow right off. And something like an insect's exoskeleton is also going to have a waterproof coating on top of it, again, created by waxes. But what's the most common form of wax that you might think of is going to be beeswax or honeycomb. And these animals are actually taking pollen and creating honey, and then they're storing that honey for later in these little honeycomb structures, and that way even if they themselves don't survive, the next offspring will be able to consume this in times of need. All right, so let's talk about phospholipids for a second, and I'm only going to kind of gloss over it here because, again, we're going to spend an entire chapter talking about phospholipids and how they role in the membrane and the membrane structure. But phospholipids are what we call amphipathic, and I know that sounds like a really big word, but what that means is that it has one region that's hydrophobic and one region that's hydrophilic. And that means that it's able to interact with both water and oil at the same time, right? And these are very important because they make a barrier. They're the biological molecules that form the plasma membrane that surrounds all cells. And again, they have a region that has the two fatty acids and glycerol. That's going to be the region that is going to interact well with, that's going to be the hydrophobic region. And then the short polar functional group, which is going to be hydrophilic, which is going to interact well with water. So the polar functional group form the head. That's the part that's water soluble. Non-polar fatty acids form the tail, and they're water soluble. And without a depiction or a picture, that doesn't really help you. So let's show you what it looks like. From here on out, you're probably going to see this depicted as one circle and two legs. So I'm not ever going to ask you to draw any of these biological structures. But what I want you to show, or to be aware of, is that we have the same fatty acid tails that we saw in the triglyceride, except there's only two of them here instead of three, and that sometimes they're saturated, like this one here, all single bonds, no double bonds, and sometimes they are unsaturated, which means there's at least one, if not more, region that is bent, which allows a little bit of fluidity, and that's going to allow the plasma membrane to be able to move about. These individual molecules are going to orient all together as a whole, such that all of the polar heads are aligned in a layer, and all of the fatty acid tails are aligned in a layer, such that we have one region that affiliates with water and one region that affiliates with oil or other hydrophobic molecules. All right, so steroids are another type of these biological molecules. And the importance about steroids is that that's going to include cholesterol, which is going to be plugged into the plasma membrane and allow stability in the plasma membrane. And when we talk about cholesterol, you may hear that cholesterol is bad, but it's actually a ratio. We have two different types of cholesterol molecules. Actually, we have several, but two major types: high density and low density, so HDL and LDL, and it's kind of a ratio between HDL and LDL in your body. That's the important part. But cholesterol is going to be found in all of the membranes of the animal cells and allow for it to give it a little bit of rigidity and more structure. And it's involved in the creation of both male and female sex hormones. When I show you the structures of the two, you'll be very surprised at the similarities. Cholesterol is found in the human brain, approximately 2% of the human brain, so it's very important for brain function. But the downside is if you end up with excessive cholesterol, sometimes you can end up with cardiovascular disease. And this is why people worry when they are concerned with the levels of the particular LDL and HDL molecules, because if you end up with high levels of cholesterol, you can end up with atherosclerotic plaques, which can end up leading to either coronary artery disease, or depending on if they end up in your brain, they can end up being a stroke. So excessive cholesterol can be bad for your body. This is what cholesterol looks like, and I will never ask you to memorize this, but I want you to see that it has got 3 hexamers and a pentamer. This has 3 hexamers and a pentamer. This has 3 hexamers and a pentamer, right? They're very, very similar, and a cholesterol molecule is easily modified into either an estrogen or a testosterone molecule, and these guys are actually able to be converted back and forth as well. You see the difference is really only here. We have an OH. Here we have a double bond O, and additionally in testosterone we have this extra CH3. That's a very similar molecule, so you can see that these guys, although they are involved in all of your sexual reproduction and development, they actually oversee some very similar processes from an evolutionary standpoint. And I also want to take a moment here to say that it is not just male and female, right? Testosterone is something that is also found in women. Estrogen is also found in men, but again, it's a ratio. So men have much more testosterone than women, and females have more estrogen than males do. Sorry if I bungled through that. All right, moving on. Let's talk about proteins. Proteins are the workers of the cell. Proteins are going to be comprised of the monomers of amino acid is the monomer. The dimer would be a peptide. After that, we get to a polypeptide, and proteins have when I say a variety of function, there's 30,000 or more proteins that do different things in the cell. We have different types of functions, but there are tons of different. For example, enzymes are proteins that promote a chemical reaction. They increase the rate of chemical reactions. That's a whole class of proteins, but there's hundreds of different types of enzymes because enzymes are very specific to the chemical reaction that they promote. We also have structural proteins such as elastin, keratin, and these are going to help provide support for the cell. So these are going to be kind of scaffolding proteins within the cell. So again, these are an overview of the different types of or classes of proteins. So we have structural proteins such as keratin and silk. Keratin is found in your hair, in your nails, also not in yours, but in scales, feathers, and horns in other animals. Proteins that are involved in motion include things like actin and myosin that are found in your muscle cells and allow for your muscles to contract and then, of course, relax. Also, all of your immune response, so your defense, are going to be found things like antibodies. These are proteins as well. Antibodies are specific to what they interact with as well. And also things like venoms, which aren't something that we as humans have, but other organisms use as defense as well, are also going to be proteins. One of the classic energy protein storage molecules is albumin. We're going to use albumin as a in the biomolecules lab if we actually end up having face-to-face courses this semester. And albumin, again, it's found in egg whites and is going to be able to provide nutrition for the developing embryo until it's able to crack out of the egg and then end up being able to consume food on its own. Proteins are also involved in signaling. So signaling molecules are able to determine self from non-self, for example, or insulin is going to be a signaling protein that's secreted by the pancreas. It's going to help increase the rate of glucose uptake into cells. And this is one of the ways that the mechanism of disease that goes wrong in diabetics is that we can end up with either insulin resistance or production of insulin that isn't functional, or a non-production of insulin that can end up making it that blood sugar is very important for these individuals. Monitoring their blood sugar is very important because they have to add insulin exogenously, meaning having to eject it themselves. We also have catalyzing reactions. So these are going to be enzymes. Enzymes are, anytime you see ASE at the end of something, it's going to digest an OSE, which is a sugar. So amylase, which is the enzyme that digests amylose, is going to be one of the very first enzymes that food that goes into the body is going to encounter. It's found in saliva and then also in the small intestines, and it's involved in digestion of carbohydrates. So amylase takes a cracker, which does not taste sweet because it is carbohydrates, and breaks it down into simple sugars, which do taste sweet. Here's some examples of some structural proteins. Again, hair, horn, and silk. These two are made of keratin. Silk is its own structural protein. And proteins, again, are polymers. They're created from a chain of amino acids that are held together one at a time by these peptide bonds. And amino acids overall have a similar structure, but they also have a variable group, or an R group, or a reactive group. So all amino acids have one side that's an amino group, one side that's a carboxyl group, and then a little hydrogen that hangs off. And then they have a variable R group that is able to react. So some R groups are hydrophobic, some are hydrophilic, some are large, some are small, some have 6 carbon rings in the center of them. Some of them are going to be split and have different regions that dangle off. A specialized group called cysteine has a sulfur in there that can bind to another sulfur, so 2 cysteine groups can come together to form something called a disulfide bond, which is kind of like a piece of Velcro in the middle of the protein that allows it to fold in a particular way. And what does that look like? All right, so this is the common format for all amino acids. A particular amino group on one side, a carboxylic acid group on the other side, and then that dangly hydrogen that I just talked about. Every single amino acid that binds to the next amino acid is going to do so by losing this hydrogen and oxygen on this carbon, and then losing this hydrogen on the neighboring amino acid. So this is not going to bind here, right? It's going to bind to the amino group on the amino acid next to it, and that's going to form a peptide bond. Additionally, what we're looking at here is this R group, and this R group is depicted just as a blue ball here. However, it has a ton of different, there's actually 20 different amino acids, so there's a ton of different variable groups that can hang off. So 20 different versions of what this can do, and it can be charged, it can be uncharged, it can be large, it can be small, right? It can be polar, nonpolar, so lots of different things that can happen there with those functional groups. And this is just what I'm demonstrating to you in this image. You do not, again, have to memorize this. I just want to show you that we have a diverse structure of amino acids. So we can have hydrophilic functional groups. These here are the blue regions. Remember, we just talked about this R region being blue. This is going to be depicted here. This one is going to have the acidic group hanging off the edge. This one also acidic group hanging off the edge. This is glutamic acid, aspartic acid. These guys are going to be hydrophilic. These guys here, phenylalanine and leucine, they're going to be hydrophobic. Again, I'm not going to ask you to memorize which is which. I just want you to show or to be able to visualize the major differences between how this might interact with a molecule nearby versus how that might interact with a molecule nearby. And this one is special. This is the one I was talking about that forms what we call a disulfide bond. So if you picture that we had this exact amino acid in a whole different part of the protein, and that protein folded, we might be able to bond this sulfur to the sulfur of the other cysteine somewhere else in the protein, kind of like a Velcro linkage or like a snap linkage between one part of the protein and the other. So cysteines are very important for what we call disulfide bonds. All right, but I digress a little bit. Just like any of the rest of these biological molecules, the monomers are created by dehydration synthesis. Or, I'm sorry, monomers are turned into polymers by dehydration synthesis. And in order to do that, again, we're going to lose one hydrogen from one side, the OH group from the other side, and that's going to have a water group. And the covalent bond is going to be resulted, and that's going to turn into what we call a peptide bond. And if we have 2 of these together, that's called a peptide. Long chains of them after that are called polypeptides, and we just know them as proteins. And that sounds like that might be it, but actually gets a little bit more complicated because it's not just the order of these proteins in the protein chain, it's actually the structure of the protein, but we'll get to that in a second. So this is showing you exactly what I've shown you a couple different times. A dehydration synthesis is the mechanism by which 2 monomers come together to form a polymer. In this case, our monomers are going to be the amino acids, and they're going to form a peptide bond with one another by losing the hydrogen here and the hydroxyl group there. Those are going to come together to form this water molecule that's going to leave a positive and a negative charge between these two, which is going to get filled and equalized here by a covalent bond, making a peptide bond. And we can make a long chain of these amino acids and create a polypeptide or a protein. But as I mentioned, it's not just the order of the amino acids that's important, it's the way that the protein is folded. So the primary structure is what I just talked about, the sequence of amino acids. Which amino acid is bound to the amino acid next to it by peptide bonds to make a long chain. But as I mentioned, it's not just your neighbor to your left and to your right that you interact with, right? Picture that you're in an apartment complex and you can interact with the neighbors above you and also below you. And so we have other secondary structures that are able to be formed when different amino acids that are not directly next to each other but still end up next to each other are going to interact. And we get specialized secondary structures like an alpha helix, for example, or a beta pleated sheet. So some examples of these secondary structures you're going to see multiple different times, and then the way that all of the secondary structures in the entire chain fold together is the tertiary structure. This is much more complex. In fact, people hold contests every year for predictions of how proteins are going to fold based on the amino acid structure, and they get a lot of different ideas, and then they do X-ray crystallography and reveal the actual result and see who's closest. And although we have a lot of algorithms, we're not very good at it yet. There's a lot of guesswork involved in which of the cysteines are going to bond to each other, for example, to create those disulfide bonds. And last but not least, we have something called quaternary structure. And this is when we have multiple protein chains that link together to be able to create a large multi-protein complex. Something like hemoglobin, for example, is going to be when we have 4 different globin molecules that come together that are going to be bound around one heme group to be able to make the ability to carry oxygen throughout the body. All right, but I digress. Here's what I meant to talk about. So here are the different levels of protein structure. This is just the primary sequence, literally just what order is every amino acid bound to in the polypeptide chain. But as I mentioned, it's not just who's next to you, right, left and right, it's also who's above and below you. So here's an example of secondary structure. This is a helix. If you picture this like a staircase, that's a circular staircase. If everyone was standing on the circular staircase, you might be able to high five not just the person to the left and the right, but also above and below you. And so these are different bonds that are held together. These are hydrogen bonds. As you recall, we talked about bonds when we talked about atoms and basic chemistry. But these hydrogen bonds are going to be fairly strong together, even though individually they're weak, and that's going to allow it to have these secondary structures or more complex shapes, which combined together create the tertiary structure. And then multiple proteins together combined are going to create the quaternary structure. So this is one globin molecule that has one heme group. If we put them together, we have 2 alphas and 2 betas, for example, or 2 alphas and 2 gammas, or however it works out. We have the heme group here that each of the oxygen molecules are able to sit on. Right, and so this wouldn't have the same function that it does if we were to completely detangle this and just have this amino acid primary structure, right? All right, so these are examples of what I was talking about, my secondary structure. We have a beta-pleated sheet. I just showed you a second ago the helix, which is like the circular staircase. But the beta-pleated sheet is more like the inside of a corrugated cardboard, right? It goes up and down and up and down and up and down and makes a sheet. And that makes it extraordinarily strong. So if you picture something like silk, which has been known for years to be very, very, very tiny in comparison to its strength, right? So it has a very high, what we call tactile strength. And that's because it has these beta-pleated sheets that allow it to have this organized complexity to give it the strength within a strand of silk. Now, just like everything in biology, form defines function. You're going to hear me say this 100 times throughout the semester. But the main point that I'm making here is that protein function is going to be related directly to the form that the protein takes. So the three-dimensional structure is going to determine how the protein interacts with other molecules around it. And that's because we have a very precise positioning of those R groups, the amino acid R groups that are going to hang off of the different amino acids. And that's going to allow them to interact with the other molecules around it in a specific way that's going to determine both the secondary and then eventually the tertiary structure. And if we disrupt that, it's what we call denatured. And we can do that by high heat, high salinity, high or low pH. We can denature a protein. And this is why homeostasis is so important, because if you figure that proteins are the workers of the cell, and proteins can end up denatured if we end up too hot, or if we end up with a pH that's too far out of the range, or if we end up with too much salt in our system, our cells can literally die because of the proteins being killed off if we end up too far out of homeostatic conditions. But I digress. Again, a denatured protein is one that has been either heated or high salt conditions, and it is going to fall back into that primary structure. I'm going to go backwards just a second. It's going to fall back into this structure, but we'll have lost all of these, and therefore these and those. And once a protein has become denatured, it's essentially useless. It's almost impossible for it to go back to its quaternary structure simply because of all of the major steps that had to happen during protein synthesis. So basically all of these different secondary and tertiary structures that we see are happening or created when the protein is being made. So it doesn't happen as if the protein is already made. All right, so let's move on to nucleotides and monomers. Again, we are going to get proteins are going to have their whole, they're going to have an entire chapter as well. Nucleotides are going to be the monomers for nucleic acid chains. We basically have 2 major classes. We have deoxyribonucleotides and ribonucleotides. Deoxyribo being what DNA is comprised of, ribose nucleotides being what RNA is comprised of. But all nucleotides are made of similar 3 components: a phosphate group and a sugar group. So when we look at DNA, we're going to see phosphate, sugar, phosphate, sugar, phosphate, sugar on the backbone on both sides. Going up, we are going to look at it like a twisted ladder. It's going to be the outside handrails are going to be sugar, phosphate, sugar, phosphate. And if you consider that the rungs of that twisted ladder are going to be the nitrogen-containing bases, which are going to pair up together to make base pairs in DNA, that's basically going to be what a nucleotide is comprised of. So this is a monomer. This is a monomer of deoxyribose, and I know that because this sugar is a deoxyribose sugar. And this base pair is going to be connected to the sugar, which is connected to the phosphate. And if you picture that if you take this whole thing and copy and paste it and put it right here as well, we would have one base pair hanging off beneath us and then a sugar phosphate backbone. This sugar would bind to that phosphate and make a long chain. However, the bases would pair to the middle because we'd also have another one running this direction. And I'll show you what that looks like in a second. But my point is that the base pairs are what are interacting with another chain running alongside it. But that would be for DNA. DNA and RNA have different structures entirely. And for a second here, we're going to talk about ATP as well, which is another nucleotide, which is created from the base adenine. So again, we have A, T, C, and G for the base pairs for DNA. For RNA, we have A, U, G, and C. And adenosine is the base pair that is the A is comprised of. And ATP is the adenosine base with 3 phosphate molecules. It's a deoxyribonucleic adenosine, 3 phosphate functional groups on the outside of it. And again, this is our energy carrier molecule. So the high energy version is ATP, and the lower energy version would be ADP. It's not going to be the only energy carrier molecule that we talk about, but it is going to be one of the most common ones that we talk about. There's another messenger molecule that we'll talk about kind of lightly throughout this class known as cyclic AMP, but again, that A here also stands for adenosine, which is a nucleotide from the with the deoxyribose sugar. All right, another set of nucleotides that we'll talk about that are not, basically what we're talking about here is everything that's not DNA and RNA, so exceptions to what I said with the DNA and RNA. These are going to be nucleotides like NAD and FAD. These are other electron carriers or other energy transport molecules that transport a little bit differently than ATP, so the energy isn't carried in the triphosphate to diphosphate. It's carried in the form of high energy electrons. And when we talk about the electron transport chain, we'll talk more about electron carrier molecules. All right, so this is what ATP looks like. So again, this is our base with the sugar, and this is called adenosine, and it's connected to 3 phosphates. 1, 2, 3. So 3 of the phosphate molecules are the high energy version. And the low energy version, again, the energy is carried here in this bond between di and triphosphate. And so the low energy version would be severed here, and it would be a diphosphate with only 2 of the P's on the end of it. So that would be ADP. Now, anytime we take nucleotides and put them together, we make polymers, right? So again, 2 monomers coming together make a dimer, from a dimer, trimer, from then into a longer polymer. And just like any of the rest of them, they're made by dehydration synthesis. 2 major types of polymers of nucleic acids include DNA. DNA is deoxyribonucleic acid. It's made of the sugar deoxyribose. And then has that nucleic acid base pair, and that's going to be found in chromosomes. Again, we have the sugar phosphate backbone on the outside and the base pairs hanging towards the center, and these are going to be responsible for carrying all of the genetic information needed for protein construction. And don't worry, we get an entire chapter on how DNA makes RNA and how RNA makes protein, which is the central dogma of biology. Each of the DNA molecules is going to have 2 chains of nucleotides. So again, we have the sugar phosphate on the one side of the backbone and the sugar phosphate on the other side of the backbone. This kind of looks like a twisted ladder. And we have the hydrogen bonds in the center between 2 base pairs, which are going to make those rungs of the ladder. The whole thing looks kind of like a double helix. RNA is made of ribonucleic acid, or is ribonucleic acid, it's short for. It makes copies of the DNA. And also is involved in creating proteins. So it's basically a Xerox from the DNA, and then also is allowed out of the nucleus through the nuclear pores and helps direct synthesis of proteins by binding to the ribosomes. And again, we'll get into that extensively, but basically that can happen either free in solution or it can happen on the rough ER where ribosomes are actually attached. And this is what DNA looks like. Again, I'm not going to harp on it too much this chapter because we do have a whole chapter on DNA making RNA, making protein. But just to give you the overview, this is 1 monomer, right? It's 1 base pair. It's a base pair G. This base pair is C. Here we have the sugar phosphate backbone, sugar, phosphate, sugar, phosphate, sugar, phosphate. Anytime you see that little yellow ball, you can pretty much assume that it's a phosphate molecule. And again, sugars are going to be depicted. This one is a pentamer, but also sometimes it's a hexamer. And that's going to make this long twisted ladder. So sugar phosphate backbone in the other direction. It's actually running the equal and opposite direction, but that's not neither here nor there for the purposes of this class. And in the center, we have base pairs. Now, as you may recall, G binds to C and A binds to T. And what's important to note here is that these are binding together based on size and based on the number of hydrogen bonds that they can create. So a simple Cliff Notes version, if you have to just remember it, is that G and C are both curved letters as capital letters. So it's easy to that those two go together. And A and T, again, these are both straight line letters, so that's easy to recall. But the real truth is that they're binding by the fact that one of these is large and one of these is small. If they were both large, they would overlap too much. If they were both small, they wouldn't reach. And in the case of A and T, they're each able to make 2 hydrogen bonds. In the case of G and C, again, one large and one small, so 2 G's together would be 2 large, 2 C's together wouldn't reach. But the G and the C are able to make 3 hydrogen bonds, and so that's why, for example, G can't bind to T because it has 3 and T can only make 2. And so that's going to have the specificity of G binding to C and A binding to T. And we're going to call it right here for the purposes of this class. So I appreciate you listening to me for biological molecules. Happy studying. Aloha.